we're the map core component of the Spark project, and that means that we're mapping data from multiple species, including human, but a lot of the data is focused on other animals. And we need to create environments where we can create a coordinate system that allows us to compare data across species. So this slide just shows the colon model for human, pig, and mouse. And in particular, what it's showing at the bottom is that each species has a rather different detailed structure, the haustra of the colon and the structure of the tenai coli. So we need to be able to essentially use high order finite element. Please interrupt me at any stage if anyone wants me to go into more detail, but we use high order finite element definitions of the three-dimensional coordinates of a particular sub-region, in this case a haustra, which we can then snap onto a center line that's been defined for the particular colon for that species. So the center lines are very, very different for human, pig, and mouse. Pig's got this sort of curly structure, and the haustra are also, antennae coli are different. So we need this ability to be able to create these almost, you can think of them as a sort of CAD-like environment, which provides material coordinate descriptions within a finite element framework from which we can then export detailed finite element models for computational purposes. And these scaffolds are always designed that are a common way of dealing with the cross-species comparisons. And so in the case of the colon, and I'll apply this to other organs in a minute, but in the case of the colon, very often the data that you want to embed or register to that scaffold is data that's being measured on a flat preparation. So experimentalists will cut along the mesenteric border, flatten, in this case, a mouse colon, flatten out the preparation, pin it out, and then do detailed confocal microscopy, for example, or whatever the technique is, to give the three-dimensional data at a particular point in that flattened scaffold. And here's some data from Martha Howard showing the vascular system within the mouse colon. And then what we do is embed that data into the flattened scaffold. So you're finding the material coordinates of each of the segmented points of those images. But once that's done, then the flattened scaffold can be then just folded back into its full 3D structure. And that's what you're looking at in the this part here, where the vascular data is now seen in relation to the overall three-dimensional geometry of that mouse. So if you then have other data sets obtained in a different way, they can all be brought into this common framework for then building computational physiology models. So here's another example, and this one for the stomach. And this is using data from the Terry Powley group at Purdue. So again, the scaffold is shown on the left. And here you need, and most of these scaffolds need multiple layers through the wall to pick up the relevant detail at different depths through the wall. So the top left shows the scaffold expanded just to show some of these layers. The middle on the left just shows how the stomach connects into the esophagus and duodenum. And then the bottom left shows, again, the fiber structure within one of the layers. That fiber structure is defined throughout all the layers, of course. And then it also shows the vascular system for the stomach, shown here on the surface of the outer surface of the stomach. And then the middle image shows the various regions of the stomach, fundus, corpus, antrum, etc. And again, the underlying coordinate framework is common for all species. So you can acquire data from one species, but then compare it to another at any given material point. And because these scaffolds are available for computation, that means we can dilate them, as you'll see in a minute, sometimes you want to be able to compare data across different states of filling of a stomach, for example, or in the case of the heart, levels of a contraction. And then on the right-hand side, it shows just various cell data sets. And this is data from John Furness in Melbourne University, where it's a different data sets. In fact, the way they open the stomach out is rather different, but we can make the model replicate the particular experimental technique they use, so that we can then take that flattened cell type data and embed it back into the three-dimensional. So here's an example where we're creating the scaffold. Initially, they're created from micro CT images in some cases, or it can be multiple sources of image data that are used to define the geometry. For the stomach, it's typically micro CT, and then that data gets registered. Sorry, that anatomical micro CT data is used to fit the scaffold to that particular data set for that particular individual species, experimental species. Then for that particular individual, they cut along particular borders in the stomach, open it out. And so what you're seeing here is the opened out, flattened experimental preparations are shown at the bottom there. And then the opened out scaffold is shown at the top. So when they measure neural tracings, and one's shown in those flattened segments, we can then we can make the flattened scaffold match the particular cross section for their experimental data, and then bring all that data back from multiple sections into that common coordinate work for the whole stomach. So this is now showing all the neural tracing for all the individual rats that have been processed, but now pulled back into that common average rat scaffold. Then, because they want to do this on multiple states of filling, we can then, at the bottom in the middle, shows a number of stomachs, individual rat stomachs from a more filled state, and then a smaller number from a less filled state, and a smaller number again from a even less distended state. So the scaffold can be made to follow that distinction in order to bring in any data that's obtained at a given level of distinction. So it's a common framework across species, across individuals within that species, and across different states of deformation for the particular. Then on the right now is just shown some data from micro CT imaging of the bladder, where we can then bring that type of structural data in bladder at the top, the urethra below, into a similar scaffold framework for the bladder. Then this time it's showing the scaffolds for a 3D heart. So on the bottom left, you can see the scaffold for the ventricles and atria of the pig heart. But again, the scaffolds have been designed that we apply them mouse, rat, pig, and human. There are slight topological variations, a number of the way that the number and the way that the pulmonary veins enter into the right atrium varies with species. So the scaffolds have to deal with that topological variation. But in this case, we're fitting the pig scaffold to data sets that in this case have been obtained with video cameras, where they've also, this is from the UCLA group of Shiv Kumar and Jeff Bell. And the patch that you see, that brightly coloured greeny blue patch, are 64 electrode patches that are placed on the epicardium of the heart, where they've measured the electrical recordings under conditions where they're stimulating the stellate, for example, stellate ganglion, and looking at the response. So we then can fit the heart scaffold to their data sets, but also fit their experimental 60 electrode patch, and then identify the material coordinates on that scaffold corresponding to those electrodes, and then record all the data that's associated with each of those electrode recordings. Those data sets are shown on the right. And then when that's all registered to the scaffold, we can, within the visualization environment, we can then show you can hover over a point and show the data set associated with that particular point. And these experiments are dynamic, so the heart's beating, so the scaffold is following the deformation through the cardiac cycle. Then I just wanted to show the way that the connectivity between organs is handled. So we map all the individual segmented images of connectivity data through a framework called AppAnatomy, which is dealing with all the metadata associated with annotated terms from these various pathways, handled within an environment at UCSD, the Crunch environment, and then all that metadata is available for mapping on the right-hand side, mapping out to flattened 2D, what we call 2D flat maps. And we have one of these for each species. So this one's showing human, but we have them for rat, pig, etc. And then all that connectivity data, which is curated by experts for the various regions, say for the bladder or for the superior cervical ganglion, etc. Each agrees on the appropriate pathways and annotations. All of that goes via AppAnatomy, that topological diagram in the middle, into the common database, and then is available for us to then map into these two-dimensional scaffolds or flat maps. Then that means we can, when you're navigating from the flat map to a particular organ of interest, you're seeing the pathways of interest between organs in the brainstem, for example, but you're then able to bring up a three-dimensional scaffold model of the particular organ you're interested in. In this case, it's showing the heart, and then interrogate points within that scaffold, for example, the RNA-seq data for particular cells that are embedded within the right atrium. And the one that's being looked at is shown in green. But you can then move across different cell types to interrogate the data associated with that. But also you can use the scaffold to take multiple data sets from different spatial locations and produce field data that then shows the distribution of a particular molecular species, how it varies spatially across a region of the heart. And then finally, because of all of this data is being mapped in order to be able to use it for computational purposes, for understanding the physiology, we're developing models of control, one shown here for the bladder and the heart. These are from published papers from, on the left from the Melbourne group and on the right from the UCLA group. And then what we do is to use a framework that we've developed called CellML, which is an XML encoding standard for models of physiological processes. And we deal with multiple standards, CellML, SPML, and then the actual process of doing a computation is defined by a standard called SED-ML, and another NeuroML is another standard for dealing with neural data. We can bring, if the models are encoded in these standards, then we can use open source, freely available software to run these models and then link them to the three-dimensional frameworks that I've been talking about, so that you can choose various models of parts of your system, pull them all together, and then run composite high-order models that bring in cellular or molecular level data from different parts of your overall anatomical framework. And so the Physiome Repository, as it's called, has about 700 models of different physiological processes encoded in CellML, and there's a similar repository for the biomodels repository for SPML, etc. So all of these models are being brought in where they're relevant, and where they're of interest to a particular investigator can be brought into that three-dimensional coordinate framework that I've talked about in order to provide an anatomical framework for interpreting the physiology of the cellular and molecular level models. And that's it. I'll stop there.
Thank you, Peter. That was a beautiful presentation. I'm just curious because there's a very small number of people here. Obviously, I'm not running it, but I'd be really curious to know what fields people are coming from, because I know people were introduced earlier, but I just can't remember who comes from what area. So just briefly, I'm an immunologist, so things get rather dynamic, but are actually very structurally, physiologically regulated. Most immunologists seem to forget about physiology, and I also do a lot of computational biology as well as being involved in the single cell atlas.
And maybe I should mention I'm a bioengineer, but very much also involved in physiology. So I originally did my PhD in the physiology department, but my original training was in mathematics and then became a bioengineer, engineering and mathematics. Aaron.
I guess just to introduce myself, my name's Aaron. I'm a postdoc with my extended Christina Curtis. My interest in this specifically has to do with my work in the HTAN project. We're trying to understand, we're trying to take into consideration the spatial locations for the different benign polyps that arise in the specific types of hereditary colon cancer that we study. And so what we've done so far is we've been able to basically in the exact model that you shared, take the colon, flay it out, and then it's covered in polyps. And so we want to be able to get information about each of these individual polyps and understand what their locations are also. So after we take a polyp, we label that location, and then once we're done taking all the polyps and then getting all the locations, then we take a picture of all of the different spots. And what we're trying to do now is integrate all those pictures together so that we can get an idea as to where everything is. And then once we have that, then whatever sort of analysis that we do on these polyps, we can take that into consideration with the spatial information that we get from those images. And so being able to develop some of these things that you've shown, like with the heart, and taking into consideration all the different places and looking at the RNA-seq profile at the different spots where the heart was sampled. That's basically exactly what we want to do with the colon. That's my interest here.
Hey everybody, I'm Tim Tickle. I work with a lot of data centers and software solutions and such for single cell genomics. I'm here to be a scribe today and to learn a lot. So I don't know if you've established a scribe yet. If you had, that's fine. But if you haven't, I'm happy to keep typing.
Keep typing. Thanks, Tim.
Hi, I'm Ellen. I'm currently working with Kati Borner's group for CCF development at Indiana. And my background is I'm the sole biologist on their group. They're basically working on mapping data to the Common Coordinate Framework. So I'm interested to learn what's possible for trying to spatially localize data. I do have one question, Peter. Everything that you presented were for hollow organs. Does this same methodology, would this make any sense to do for solid organs like kidney?
Yes, no, these are all solid organs. Well, sorry, I see what you mean. If they've got ventricles, yeah, no, absolutely. We are developing a scaffold for the kidney. There's absolutely no limitation in terms of the shape of the organ.
Great.
We're also doing them for ganglia, because very often you need to be able to bring in multiple data sets for stellate or nodose, etc. ganglia. Lucy.
Hi, I'm Lucy from NIH-HCA National Heart and Blood Institute, and I will just be the observer for the meeting here. Okay, thanks.
Okay, thanks.
So I guess one of the things that I was thinking about, in terms of the how might we sort of questions, had to do with how do we make this a dynamic sort of model where we take into consideration the changing of this organ throughout time. So how does this look from a young organism versus an older one, and how does that change? And maybe even how is it perturbed through different drugs? Those were some of the things that I was thinking could be important here. Yeah, and I'm new to this also. Maybe this is a simple ask, but how might we gather important spatial information data from these things that are generated? Those are the kind of questions I was thinking about.
So I think in terms of growth from into adult form, I mean, a lot of the scaffolds we develop are based on adult individuals, but we have done work on looking at the embryonic growth of the heart, for example, and we can certainly handle the parameter changes that redefine the shape of an organ through growth through time, providing it doesn't have a topological change. Then it becomes harder. Obviously, in the early stages of embryological growth, you're getting major topological changes going on in the organ systems. So we haven't tried to tackle that yet, but certainly once you've got a fixed form, a child heart, pediatric heart, for example, then that can be tracked into adult, if you've got the data, can be tracked into adulthood, and the scaffolds can that growth process through time.
And then is there a way to test this model with different growth parameters? I'm sure that there is, but I feel like that could be an interesting addition to these dynamic models.
Yes, the case that I mentioned where we're doing it with the heart, which is it's an embryonic. We're looking at the folding, bending, twisting process that goes on in the embryonic mouse heart. We actually measure all the cells. We do an analysis where we look at every cell, and we also do micro CT images on the different stages of growth, so that we fit the scaffolds to the micro CT images, so we know the overall geometric changes that are occurring during growth. And because we're doing the cell analysis at each stage, we also know the change in shape and orientation and size of each cell, so we can then relate cellular level growth to the overall organ tissue level growth. What we haven't done yet is to do all the proteomics to understand the pathways associated with driving that cellular level growth. But the framework could certainly handle that if the data was there.
Peter, I know I said I was observing, but I'm really curious here. This is really cool stuff. Does this, when I've set into topics about CCFs, one that I set in on was about lung and how difficult that might be because of the branching. Could you comment at all maybe on maybe the feasibility of lung and how you would approach it?
You're talking about branching of airways, for example?
Yeah.
So we have models where we can, maybe I can, well, actually, while we're talking, I'll just grab some other models to bring up. But we do have models of the lung which have all the airways defined.
Whilst Peter, you're hunting for that, Toby here, and lovely to hear this rich discussion. Two thoughts I had for you. Well, one was a question. I hope everything's going okay. Second thought is, are you capturing things in the Google Doc? I think I've been watching that, and that seems to be a rich hive of furious typing activity. So thank you for that. Third most important thought is, do take a break. You know, virtual work is just exhausting. So stroke the dog, go for a walk, have a cup of tea, whatever is your thing. And but if you've got any questions, I'll be in the background and can respond to anything. The key thing is capture those things in the Google Doc. Sorry, Peter, didn't mean to interrupt you.
No problem. I'll just bring up these lung images, and then, because it's a demonstration of pathways, and then we could take a break.
What a good idea. And just agree with yourselves a time to come back and reconvene, perhaps at 10:02.
So can people see the screen now?
Yeah.
We can see your cursor, we can see your browser page at the moment.
Oh, there we go.
That's pretty.
So here's an example of a, these are all 3D, but I'm just showing 2D images of airway trees, and there's also vascular trees and connected into models of alveoli, and the detailed, in some cases, detailed capillary networks. So the airway branching trees are embedded in the scaffolds, such that you can then solve the flow equations of the airways and then link that down to oxygen exchange at alveolar level. So the scaffold frameworks have or the common coordinate frameworks have this notion of a series of embedded coordinates. So the airway, the definition of the airways is given in material coordinates that are defined relative to the lung lobe that they're in. And those lung lobes are all defined relative to the overall lung geometry. And the lung is sitting in a torso, so it's defined relative to the coordinates for that torso. But each one is obtained via an embedded coordinate process. So you're starting with the whole body and then working down through the torso, through the lungs, through the airways, down to particular points within the airways. So there's and as any one of those moves through breathing, for example, then that's been tracked within the coordinates are tracking the movement of those points within the host organ.
That's just cool. It blows my mind. That's such cool work.
So Peter, that's very beautiful. I'm just curious, how can this be applied to organs like the spleen or lymph nodes, or things where things are actually much more dynamic? They're very highly structured, but they are continually remodeling and reshaping as immune responses are occurring, and I think that becomes it maybe it's a really big problem that's beyond where the technology is now, but trying to understand, there's actually great people in Auckland, Bob Dunbar and Gib Boyle, engineers, immunologists / engineers have been working in this area. But I'm just curious as to whether any thoughts there, because of course we're getting lots of single cell transcriptomics, lots of spatial genomics information coming down, and it could be very powerful to be able to build some type of structure. It's just the coordinates, as you move organ to organ, or time and space are going to change.
Yeah, the work I'm showing here is from the Auckland Bioengineering Institute, and Gib Bogle is part of that, and so I know his work well on the lymph nodes, and it's the same. It's within the same framework, except Gib is primarily using agent-based models for the interactions, and I think we've not yet really handled the problem of rebuilding the topology of an organ based on those interactions. That absolutely needs to be done, but we're not quite there yet. But there's no reason that the frameworks couldn't do that. But it is the complexity is simply that you're having to continually refine the host anatomy based on the interactions going on within the organ.
I mean, I think in the long term, it's going to be very, very important when we try to understand pharmacology in the context of physiology, particularly from an immunological perspective. I'd be happy to be in contact with you and Gib and see where we could start to think about it. It's a big, big problem, but I think it could have big impact, particularly on, the immune system is very extensively obtained a lot of funding from the HCA area because of its role in disease. Yeah, I think it's something that could have a real impact, but perhaps I could take it offline and talk with you and Gib at some point.
Yeah, Gib's currently in lockdown in Chile, I think, at the moment. But, yeah, no, absolutely, be very, very happy to do that. I think Gib's also had some interactions with Ron Germain and at NIH.
Yeah, Ron's been doing very beautiful three-dimensional reconstructions of tissues. It's amazing stuff. I mean, yeah, it'd be lovely to see how you can integrate together.
And there's a guy, an immunologist here, Rod Dunbar, I think, is also
I know Rod
working
well,
with. Right, yeah. Do we want to take a break?
Sure.
Yeah.
Okay. And then what do? We come back to this chat room, or we go back to the main? Come back here. Okay.
I think we
go.
can.
What? 10 minutes?
Well, I think we need to start. A good idea to see what these questions we want addressed is and what recommendations we have going forward in terms of moving the field forward, because I think it can have a real profound effect on the Atlas related projects.
When we come back from the break, is it breakout session part 2 at that point, or is it still breakout session part 1?
It's breakout session part 2 at that point, and so you're sort of thinking about, from how I understand it, from how the last one went that I was in, just thinking about sort of picking something that is a problem and thinking through solutions or how to move it forward, as the gentleman previously said.
Yeah. Okay.
How long are we taking off, Peter? So we just have an idea when to come back?
Do you want 15 minutes? So back on the hour.
Sounds good. Sure.
Okay.
Sounds good. See everybody.
See you.
Cheers.
Cheers.
Peter, if you're going to stay online, I just have a quick question, maybe to understand a little better. But I don't want to bug you if you're doing stuff like checking your email.
No, that's fine. I've got a cup of tea here, so I'm good. Yeah.
Just very quickly. So I have a background in computational biology and software engineering, and I sort of sit between those two worlds. But I've never really worked in this space here at all, and so. Excuse my naivete, but you seem to be very expert in this, and it's inevitable. So for the lung, I really appreciate what you were mentioning as far as the branching goes, and the related to larger things hierarchically. It makes a lot of sense. I was under the impression that, like, eventually as the branches get smaller, there's a level of stochasticity there that's sort of hard to model. Is that true with that organ? And if so, like, that's sort of, I think, the root of the original question.
Right. Yes, I think the way that we deal with that is we often will define the large scale vessels or airways, whatever it is, of a tree in terms of real data for the 3D structure of that particular, those branching components. And then you reach a point where we switch to algorithmic generation of the smaller components of the tree. So you might go down through 3 or 4 generations based on actual data for an individual. And then you switch to being able to algorithmically generate all the way down to capillas or alveoli, where the algorithm has built-in probability distributions based on data that give you the variation of branching patterns and diameters and so on, so that you're then able to generate a sequence of models from a random number generator that picks off probabilities from those probability distributions that are built into the algorithms. Mm-hmm. So a lot of the computational work does try and develop some standard error around predict outcomes based on multiple runs with different choices on from those probability distributions to account for the variation that occurs. I mean, I think it's still a very limited process that I think there's a lot of work to be done in terms of really capturing the huge variation that occurs in all sorts of aspects of biology or physiology. I think one of the really interesting questions for me is, here I've been talking about a coordinate framework, but there needs to be alongside that a mathematical framework that's also multi-scale and multi-physics, multi-organ. And the way we do that is through something called, where you have to go to energy-based approaches. The only thing that's common between, every organ in physiology involves every discipline you can think of. You have electrical, mechanical, chemical, heat, etc. Any given physiological problem involves all of those coupled together. And that means that the only thing that's in common between those different physical systems is in fact energy. So you need to formulate your equations in terms of energy. So you stop thinking about a voltage being an electrical concept specific to electrical engineering. You start thinking about joules per coulomb, which is a volt. Likewise, in mechanics, you don't think of newtons, you think of joules per meter or joules per meter cubed for a kilopascal, and so on, joules per mole, joules per unit entropy for temperature, and so on. And that means once you have a common mathematical framework, and we use something called Port-Hamiltonian theory, which in a lump parameter sense becomes a well-known field called bond graphs, which is about energy transfer. But in the continuum, larger scale sense, involves a discipline called Port-Hamiltonians, which is quite a rigorous mathematical framework for dealing with the partial differential equations of physics across different disciplines through a common energy framework. When you have that, you can start to ask questions like, when you couple 2 cells together, each cell may be and this relates to control systems formulated in terms of that energy framework. But then you can say, if you take an individual cell and measure its properties, it's often very different from its neighbor. But when they're coupled together, they behave in a very complementary, robust fashion. And that poses a really interesting question mathematically as to what is it about the parameter variation, what appears to be a stochastic parameter variation between components, which could well be part of making a system, an integrated system, much more robust. So I think there's really, really interesting questions that we're only beginning to think about in terms of the relationship between stochastic variation and the robustness and ability to adapt of a physiological system. And it's a very long answer.
No, it's really fascinating for you to walk through all that, and then suddenly it sounds to, at least from what I'm hearing, you start to think, from cell to tissue, or at least paracrine signaling and these kinds of things, because you can model it there at that point, because you establish the robustness that you're talking about, for instance, that sort of comes with this kind of modeling.
Yeah.
But yeah, so that's super helpful for grokking it, and I know there's so much more to know about it, but thank you. I appreciate the description.
No, you're welcome. Pleasure. Are you still there, Mark?
Yeah, I'm here. Yep.
Are you based in Oxford? I can see the Sheldonian behind you.
I am interested. Of course, obviously not exactly my bedroom at home near Whitney, but yeah, no, I'm Oxford based. That's correct.
Yeah, I spent 7 years there, so I did my DPhil at Oxford, and still have strong links with people like Guinness Noble at Oxford.
Right. So you were in engineering or math?
No, I was actually in physiology at that stage. I did my DPhil in the physiology department.
Oh, okay. DPAG. Is it
DPAG, yeah. I work quite closely with David Patterson, who's the head of DPAG.
Yeah, Oxford is a vast place these days. It's grown massively since probably you were there.
Yeah.
Half the DPAG is moving up the hill to near where I am on the Old Road campus, which probably didn't exist when you were around.
No.
That's next. What used to be part of the Churchill site.
Yes,
Churchill.
I mean there was always that separate Radcliffe site and the one right up the hill, but not Old Road.
Right. Okay. Yeah. The JR. Yeah. This JR. They're long since ran out of room on JR, so they built a large new campus on what's called Old Road these days.
Yeah.
In fact, half of DPAG is moving up to the Old Road campus. They're building a big new regenerative medicine building.
Oh, okay. All right. David Patterson comes down here to do a lot of his experimental work, because we still do quite a lot of our large animal work. It's quite difficult to do in the UK.
Yeah, well, it's probably even harder in Oxford. I fail to be aware of anything other than a few primates in the animal facility, or anything other than mice. Certainly no pigs that I know of, unless they're hidden away somewhere. I suppose that probably people go down to Pirbright these days, the Institute of Animal Health, if you wanted to do larger animal stuff. But that's good hours trip in the car.
Yeah. Mind you, quick air flight to Auckland.
I would go do my experiments in Auckland any day over driving down to Pirbright, but
Do you know Carol Robinson? She often comes down here with David as well. She's heading a new biochemistry institute.
Oh, okay. It doesn't ring a bell, but Oxford has has over 1,000 faculties. So in the medical science division, so crossing paths with people can be one of those issues when you have a large faculty spread between 3 different sites.
Yeah, actually, Rod did his immunology at Oxford, didn't he?
Rod did, yeah. Rod was at the WIM, which was on the JR site. Yep.
Yeah.
With Enzo, I think. Oh no, he was Alan Townsend, maybe. Can't remember. No, Andrew McMichael. He did it with Andrew, I think. But yeah, unfortunately Enzo passed away. But yeah, I visited here a couple of years ago.
Yeah. I was very interested to hear that you knew Gib. That's great because his wife is it Chile or Peru? So it's actually Peru, sorry, not, well, no, they're in Chile. But his wife is South American, and so they're spending half their time in South America now.
Oh, okay.
Because they've got a daughter that's in Chile.
Oh, I see. Right. No, I didn't know that. I haven't seen Gib for a few years. If I see his papers every so often, so he's one of there are very few people who does what he does in the world. So
yeah,
if you're interested in the area, you have to know
him. Yeah.
Yeah, I mean, I like him. I mean, most of the stuff I do, well, not most. I mean, a proportion of the stuff I do is ODE-based model. I mean, sorry, agent-based models. We do OD models as well. But we've become very interested in the physiological translation between mouse and man. And in immunology, 90+% of the experiments are done in mice, which are not a very good physiological model of humans. I mean, actually, molecular level, they're fine if you want to know about transcription factors, but they are extremely concerning at a physiological scale, because most people do the experiments, they add like 500 times more stuff into the mouse effectively, and then don't realize the physiology of a mouse is not the physiology of a human. And then they magically don't scratch their heads when nothing translates to human medicine. Well, a mouse is physiologically not a human.
Yeah.
And what Rod does, of course, is the human tissue.
Yeah. I mean, what is quite interesting, the work we're doing on multiple organs and different species, the fact that, you know, very often even though the shape of the organ is very different and its position within the body obviously is different, as soon as you get down to tissue level and down to obviously cell molecular, everything becomes common again. So it's, I suspect, a framework which can bring all the data from different animals together could help bridge some of those mouse to human issues.
Yeah, no, absolutely. I mean, I would reckon probably 50% of drugs, their actual failure is not because the receptor isn't there or something like that, because people would pick that up fairly early on. The problem actually is that physiology doesn't translate between the species. And nor does disease either. People in mice knock out a gene and get a disease. In humans, they walk into the clinic. I've been trying to say this with COVID-19, you know, and because we get involved in stuff, being an immunologist. I keep on stressing to people that they don't come into the hospital until they basically have pneumonitis. But they come in because they've got breathing problems. So anything which is, you know, delivering a drug to a healthy lung is not equivalent to delivering drug to a lung which is full of liquid.
Yeah.
And you know, and it's all this, you know, and your mouse model is great, but it's not going to tell you how to treat patients when they come into the ICU. But yeah, I think, you know, immunology probably is the biggest step to go. It's time, I think, for us to. I managed to get my tea fairly fast. Yorkshire tea as well.
I'm drinking English breakfast tea here.
Well, in England, because I lived in Yorkshire for a while, it can only be Yorkshire tea.
Yeah.
How are they different? I'm drinking Twining's English breakfast myself, but how are they, as Yorkshire, different?
The Yorkshire Tea Company. They're probably, it's subtle, to be honest.
Gotcha. Now I understand.
PG tip.
I find tea in England tends to be a lot stronger than tea elsewhere, I think.
Yeah,
I don't
yeah.
know, the tea
God didn't taste.
seem to have more. Yeah, yeah.
I would have thought the New Zealanders would have liked the English taste in tea.
Yeah, no, I mean, we drink a lot of English breakfast, but it's still a little bit weaker than the English breakfast I drink in England.
But when I go to the USA, I always have to bring tea bags from England.
Yeah, yeah, yeah. We're fairly nervously watching the numbers today and tomorrow. I think are fairly crucial for us in terms of COVID-19 because the country has clamped down about lockdown 4 days ago, but we closed the borders about a week ago. Well, they're really probably only today they're totally closed. But what we're seeing is a peak from travel related. And we're just nervously watching the development of a new peak from the transmitted COVID. So the travel-related peak is dropping off, but potentially the transmitted one may be taking off.
Just keep Gibbo back in Chile then.
Yeah, well, it's probably no better.
It's actually seemed to have come down a little bit today in the UK. So. But I think it will be a long 12 weeks or so. You know, they've probably flattened it off a little bit, but it's going to be quite a long tail. I mean, they're talking
and I suspect
6 months.
And it's going to be a long time before international travel really gets going again.
6 months, really?
Yeah, that's what they're saying. Well, but not back to normal probably in 6 months. Well, what will happen is as people relax, it will just rebound again. I mean, the States is probably even worse because, you know, you might be able to somehow repress in New York or Chicago, and then when you de-repress it, they just will pop up in Tennessee or in wherever. And then it will get back to New York City again or Chicago again, and then it will go up again. I mean, all the models, mathematical models show very nicely that you can get these rebound spikes.
I wouldn't be surprised. I mean, we're not even testing correctly right now, so you know, no leading the charge here for sure.
Now, I think one of the interesting questions here is going to be: do you ease off on the lockdown, but keep all the 60, 70-year-olds at home in order to then gain a bit of immunity in the population, and then clamp down again and sort of do a whole sequence of clamp downs and release?
Well, it all comes back to parameter values and how confident you are in those parameter values. Actually, fairly small value changes in some of those models can make a very big difference to predictions. That's the one thing people sort of they take them with faith, but the
R noughts have been going up a bit. I noticed it was thought to be a little bit over 2, but it's now closer to 3.
Right.
So what we need to go back to the other meeting room now, do we?
So I think that there's breakout session 2 on the green.
Yep.
Because how might we? I guess we need to figure out what the really succinctly what are the questions we're addressing. I think there's some of those questions up above, and then how do we address them? Because we want to come a relatively small number. We want to come back with some really clear recommendations. And how that would impact on the various projects out there. At the moment, computational physiology may seem like a niche, but I think actually gotten right, the impact is massive.
Oh yeah, I think that people were just waking up to the needs that this team represents. So.
No, I'm sorry, I'm on the call. I'll just have it later. Oh. Question about whether I was going down for dinner. It's 8 o'clock.
Breakfast time here.
Still lunchtime here.
You're making me hungry.
Sorry.
No, it's fine. Go ahead.
I know it's not the most like entertaining thing to see somebody eat like while we're on like a conference call, but you know, usually we would have like time to like go get lunch.
Get used to it. We were just discussing whether it would last 6 months.
Yeah.
What's your veggie garden like? Can you grow your own food?
What was that?
What's your veggie garden like? Can you grow your own food?
Oh, no. I don't have any of that. I wish. That'd be fun. I tried to grow spices for a little while, like some cilantro and basil and thyme, but they all died.
They had a thing on the BBC News this morning that all the seed companies have run out of seeds. Because everybody's like gone on this, like they were with toilet rolls a couple of weeks ago. People have gone this mass like panic buy of seeds. I bet you 90% of them will never be planted.
Yeah, I have this fascination with like gardening and robotics and things, and so I have a automated indoor automated hydroponic system in my office, and that seemed a little silly to have, but now, man.
They come back and it's stolen from your office.
So fresh herbs.
I was looking for flour, like to bake bread, right? And then I went to the store, and that's one of the things that's just never stocked. Bread flour, all-purpose flour, barely ever find it.
Because the bakeries have closed. Yeah.
Everyone's practicing their sourdough making skills now.
Oh, have you got a good recipe? Because I so need a new starter culture.
Now we know where all the flour went.
You cannot get it. I live in Ireland. You cannot get flour for love nor money.
Yeah, you can't here either in the Midwest and United States. It's all gone, just like the toilet paper.
Yeah, it's the same in the UK. There was no probably we nicked what was available in Ireland. There's none in the Greater British Isles, or probably Europe, for that matter, as you're still a member.
Oh, yes. Listen, I didn't want to interrupt. I'm so glad you're all giggling. That seems like a very, very good result as far as I'm concerned. I just wanted to check in, saying you've probably got about 55 minutes until we were going to formally close this part and drag everybody back into plenary, where we'd literally do a little wrap up, tell everybody what we're going to do tomorrow, but also share a gallery of all of the outputs. And I suppose that's my second point, is that I'd be really keen for you to continue to contribute to the Google document that you're working on, in particular, where you think that there might be appetite for or room for cross-consortia collaboration in this particular area. So because what we'll be doing is dragging that blue section out where you've generated potentially some nice and interesting areas for potential collaboration, and we'll share that with everybody so that everybody can see, oh, there's an interesting thing that Aaron and Mark and Ellen and Tim and Lucy and Peter started to initiate. So do take a moment. You've got a good 55 minutes. It'd be even nicer if that was 55 minutes spent giggling, because that sounds very, very positive indeed. But if you also have a really productive output and you can capture that in the Google Doc, that would be even nicer.
So I'm thinking about
any questions? Sorry, Mark.
No, sorry. I was starting to think about into the blue area, but a little bit.
In that case, into the blue sounds good, and I will get out of your hair.
So the one thing I wanted to say was, I know it says problems we think we can solve by collaborating. I think also one thing I'd just like to, I think would be nice to capture is problems that, you know, where we can engage or bring stuff in with the bigger community. So I think actually people should be really excited about what's been shown and what we can do. And I kind of want to make sure people can catch that and go, wow, you know, if I could access this and utilize that, that would really impact on what I do. But I'm not, you know, at the moment, computational modeling has a habit of sort of seeming very niche. People haven't quite grasped the excitement yet.
So Mark, why would you say that people haven't really grasped it? Is it mostly just because it doesn't seem accessible to anyone other than computational biologists?
I think it sometimes scares them a little bit.
Yeah.
So I think biologists are they've gotten just about used to bioinformatics. Physiology was probably something they did in their first year in a biology course. And it's a sort of a big jump. You know, I would say even metabolomics, very hot at the moment, right? Mm. But actually, until a couple of years ago, everybody was scared of it because the last thing they remember was being tortured by the Krebs cycle on their undergraduate biology exam. It's the last time they wanted to ever remember it in their life. And I think physiology has a little bit of that. It doesn't mean there aren't some great physiologists, but I think the people doing the genomics down at the single cell level, I think maybe they like the content at a big level, but there's kind of, they don't quite, maybe not enough confidence yet. Maybe that's the word, you know, confidence they could engage with it, confidence that you could map physiology back to the single cell genome.
Yeah.
I think, well, one of the things we found in the excuse me, in the Spark project is that a lot of the experimentalists that we're dealing with don't have much background in maths or computational work, but if you can present their data to them in a way that they can visualize spatially, they begin to understand what computation could offer, I think. So it's really, I think the process of mapping and visualizing is the first step on that path to then slowly bringing in more modeling to help understand function. But the starting point, the best starting point, I think, is the visualization of the data in the 3D environments.
Yeah, I'd really like to double down on that modeling idea. I think that, you know, a lot of us study different diseases and being able to visualize it using this as a model and being able to set up whatever parameters people think are important would be, you know, really, really good. And coming up with a tool so that you can share that, so that it is readily accessible, so that people can see it and understand it by seeing it. I think that's something that we probably can do, and something that we can develop cross consortium, I think.
Yeah, I think the tool is going to be tools that make it much easier for people to feel that they can engage, I think is going to be key. Rather like how in the last few years, Surat and some of these other things made single cell biology more accessible to people without a degree in statistics.
Hi, it's Deanne. I know I'm late. I had other things I had to do, and I wanted to come to the session, and I missed the first half. But I was in the developmental biology temporal development section earlier, and one of the questions that they had was a common coordinate framework for the dynamic development of embryos, and how you compute across that. So I just wanted to add that to this discussion. It doesn't have to be talked about right now, but maybe to the ideas that people might have, because that kind of goes along with changing organs. Even though the heart is the heart, it changes structure dynamically, and then the cells change positions dynamically, sometimes from one end of the embryo to the other over time. And how do you compute across that?
And to that end, not only do you have embryonic development, but you also have cancer development.
Yes.
To be able to model that too is going to be an important thing, especially for the different groups that are involved here. Got Cell Atlas being one of them, Human Tumor Atlas Network being another.
Mm-hmm.
And the other thing is, how do we capture heterogeneity, right? Tumors are incredibly heterogeneous. Humans are pretty heterogeneous. We talked earlier about how any immunological organ is heterogeneous in time and space between people, even though the fundamentals are the same. You know, do we have, you know, a central, you know, tissue? You know, how do we deal with this, that sort of stochastic nature of biology?
I know that there are models, agent-based models, for modeling tumor growth. It usually goes down to like a single cell, or what people consider to be a DEAM, which represents, I think, 10 cells. It can represent more than that too, but, and then, you know, they let it grow and see it develop, and it creates this 3D spheroid thing. And then you can adjust the parameters, adjust the amount of selection, and it helps you look into the amount of heterogeneity possibly there. But I haven't seen it done on like in like an actual tissue sort of model. It feels more like looking at an in vitro model or an organoid model, rather than like what we're trying to do here, where you have an actual organ and trying to see how it develops from a single cell or something like that. Not a single cell, but I guess from a tumor that would be a single cell. So I know that things like that do exist for tumor modeling. Which I suppose could be incorporated into something here. I don't know exactly how, but I'm sure that it could.
I examined somebody's PhD on that, so I certainly know there's agent-based models of polyps and things like that certainly have been developed. Absolutely, there are different things. As one of Rod's poor students also got the end, got me examining him.
Nice.
To add some complication to the conversation, another thing that was discussed in the earlier temporal analysis is in terms of computational physiology was how to join animal models and human models, both in the single cell context and the anatomical context along embryonic and growth. So how do you say that this particular part of the heart in mouse is equal to that in the human, and have that temporally go along the same side along with the cell developmental stuff? So that's a very interesting problem.
Yeah, I think that one, I think, is within the scope of the scaffolds, because we do have a common coordinate system across the scaffolds that get morphed into the appropriate species. So you are able to bring data from one species back into a common material framework that allows you to compare. So that one, I think, providing there's no topological change, providing you're just essentially morphing a given topology into this new shape, that species, bring all the data into a common framework.
Has it gone? Sorry, you were breaking up. Does it go back towards the developmental side, or is it pretty much on by the time the organ's fully finished?
That was largely dealing with adult organs, but I was mentioning that we have done a little bit of work on embryonic growth of the heart, looking at bending, looping of early development of ventricles in mice, and that is that's the relatively easy part of heart development. It gets a lot more complicated later on in development when the second heart field comes in, but we can certainly handle that early stage of development by tracking what the cells are doing, individual cells are doing, and by tracking what the overall tissue is doing through multiple micro-CT imaging at different stages.
So one thing we were discussing a little bit earlier was the concept that mice and humans are physiologically incredibly different creatures. Actually, the molecular level, if you do molecular biophysics, which my lab sometimes does, so we need to go down to that scale. Actually, the difference between mouse and human, yeah, there are differences, but they're not particularly large. They get a little bit more different at the single cell level, but generally similar cell types have similar functions between humans and mice. It's when you scale that to the organ level and beyond that, you know, blood flow in a human is very different from blood flow in the mouse, and that's basically physiology, which is why pharmacology is different between a mouse and a human. Immune trafficking, you go down a long list being an immunologist, but the same with heart. These differences are, as soon as you scale up to the size of a human, you just simply can't take a mouse heart and say it's 1,000 times bigger. And so actually this stuff, I think, becomes essential if we're going to take the single cell atlas and or human and mouse disease and understand the translation between them. But that's my own opinion.
And once you're up to that organ level physiology, we know a lot about the relevant physics. And so you can actually fairly easily adapt to different anatomies. And so blood flow in a human, blood flow in a mouse, the differences are associated with well understood physical processes. So modeling can really help at that stage.
Thank you.
So I guess we'll come to the question to address is, maybe everybody could go with one question. I don't know.
I mean, it sounds like we're all trying to talk about how to model change, basically, how to model the dynamic nature of organ growth or organ dysfunction or, you know, organ development. And that could be either normal development or abnormal development, namely cancer. And I know that we're trying to talk about this in relation to mouse models. So I mean that could be one way to do it. Might be the only way.
I mean, my question would be: can computational physiology be used to understand the translation between the mouse single cell atlas and the human single cell atlas?
And developmental atlases as well, although I'm not sure. I guess the developmental atlas falls into the human cell atlas.
But there's a plethora of mouse. There's a vast amount of single cell mouse data out there. It's almost like you can't publish a paper now without doing it.
Did we talk about? Sorry again because I arrived late. Was there conversation on spatial mapping as well of single cells? For example, you know, how to map a spatial map of single cells back to the actual physiology and the anatomy.
Do you mean the changing parameters or changing behavior of a cell in different regions of a tissue, or do you mean spatially mapping within a cell?
So a challenge would be if someone took has a kind of idea where they've taken the sample from in a heart, and then they have a spatial representation of that piece, how people are mapping that back onto the organ. I'm not familiar how people are doing that, if they're doing that at all yet. Like, because you have a segment that is from part of the heart and it's been taken out, and then it's deformed slightly when the tissue goes on to the spatial technology, right? It's a piece of tissue at that point. How can that be mapped back as the collection of, let's say we just mentioned earlier, like things like 10 cells or something? How is that mapped back onto the anatomy? or how could that computationally be mapped back so that you don't need to, so that this could be a collection of spatial samples that could be then mapped directly back onto a computational idea of the heart, like a morphing or a, you know what I'm saying? Yeah. Do I make sense?
Yeah,
Yeah.
I think that the key there is having fiducial points. So if in the small sample that you're making those observations on, you've got fiducial points that can be identified, then they can be referenced back into the organ. I mean, it might be a reference based on blood vessel location relative to certain blood vessels or airways, or some feature of the anatomy that allows you to spatially identify a fiducial point that then provides the, if that can be annotated, then the 3D coordinate systems can essentially morph that information back into the common framework of the organ.
There's been discussion of collaborations and collections of large-scale tissue and single-cell resources for different tissues and across the anatomy, for at least in pediatrics and development. And there isn't yet an idea of how people would go about trying to re-register the, as you were just mentioning, having fiduciary points is a great idea. But how to remap that back onto a model of an organ that could bring in all these different samples at the same time.
What is also clear is how much information you need to do that, right? So essentially you want to gather the minimum information necessary to map back. Now, each individual cell might have 30 or 50,000 features, whatever. I mean, it's probably 3,000 or 5,000 these days. But then, you know, when we're doing the I don't know, I use CellDive and I use LightSheet, you know, when LightSheet, I don't, wouldn't go more than four antibodies at once, maybe CellDive, we could go up to 20 or 30, how do we then, and where can we maintain an integrity back to the points and the necessary to drive the physiological model. There are some things that may be easier, like, as you nicely showed, the blood vessels or the neurons, because they're very differential gene expression profiles. It's going to be relatively easy to map that backwards. I think it's going to be much more challenging to map fibroblasts populations. I think about this a lot because one of my human cell atlas project is the joint. So we look at what's called the DIP versus PIP joints, and how they're physiologically different.
So with regard to the minimal information needed, I mean, I think that, with regards to like a project that we could like do or something to work on, it sounds like we would probably need to come up with the model organ, and then from there, so yeah, that's one point, a model organ to work on, and then also the other thing was, I mean, to come up with like an average of all of these things, to come up with a scaffold to work with. In order to look at this dynamic range of sizes and things, would it be useful to look at MRI images or like CT scans of just hundreds of people and just whichever organ we're interested in, get an average of that first, and then move forward with like single cell sequencing on a couple?
Yeah, so absolutely. I want to just comment on this because Kati Borner's group has a couple of things addressing this right now. One, they're developing a registration user interface is what they call it, but basically it's getting a 3D reference organ based on CT or some sort of image data. And then initially when you're collecting the sample, like the tissue sample, kind of getting a gross anatomical viewpoint of where that sample came from on the reference organ, and then recording those coordinates, provided you have some sort of way to orient your tissue sample when you're collecting it. so that when you do have your assay data, that you can bring it back to to where that tissue sample came from in the reference organ. So that's one part of it. And then to sort of address the variability, human variability issue, there's a tissue mapping center at Vanderbilt University with Jeff Spraggins right now, and Bennett Landman, who is taking this huge repository of images, CT images and MRI images from what's called an image view VU repository, and they're looking at, they've got a little bit of funding from the HubMap group to look at human variability, and they started with 550 and then 500 scans, and they're trying to find out what sort of, for kidney, what the consensus would be and how much variability there would be for just kidney, for instance. There's been a lot of discussion in terms of how do you choose from the millions of CT and MRI scans that they have available to them. And so there's been a lot of discussion about how you pick from that repository of images so that you're actually looking at, for HubMap, for instance, we're wanting healthy human organs. And so to find images that have already been taken from the medical community, you know, scans of people and patients, typically when you go in, you're not healthy. If you're getting a scan, you're not always healthy. But if you can find a person who maybe has healthy kidneys, but maybe had gone in for something else, and the kidneys weren't as affected, you'd be able to determine that. But that's where all the metadata comes in. And I was in a metadata collection group earlier this morning. And that's another one of the discussions is for clinical metadata, they don't seem to be capturing a lot of the spatial information that would be necessary to kind of bring things back. I think both of these groups could really benefit from each other, you know, in terms of being able to, when organs and tissues are collected for this kind of work, to actually gather more spatial information, like landmarks of where that piece was taken from in the organ, because in a lot of cases we might have you know, like molecular data that's been collected, but we don't really have a good sense of exactly where from that organ it's been taken from. So then it's hard to map that back. But anyway, those are two efforts I know in HubMap that are going on in Kati Borner's group. And if you're interested in hearing more about that, I would definitely contact Kati, because she would love to put this out to more people. What we are having problems with is actually getting reference organ information. So we've been currently just using the Visible Human from NIH, which is a fairly old set of scans, but she's actively looking for other sources. And so that's just a little bit.
I don't know what's wrong with college students.
Good question.
But it may require going back. I mean, I don't know what it's like to people in the autopsy department, but when I've had tissues before, they've been from, you know, discarded transplant material.
Yeah.
Maybe Peter, you give. I know that Rod and Gibb seem to get hold of human tissues and do some really beautiful work. So I don't know where you are in terms of reference tissue in imaging in Auckland.
I don't think there's any sort of major effort to provide a tissue database. There is for the brain. Auckland has a very substantial tissue biobank for brain tissue, but not for other tissues as far as I know. So it's more on a one-off basis, tissue obtained from friendly clinicians or hospital collaborations.
I mean, it's somewhere where Wellcome Trust or NIH could come in. My guess is that because these are going to be very difficult to get a number, but we may need to decide what that number should be. But getting updated reference tissue using more recent technology, I think, would be invaluable as a proper reference set. Oddly enough, it's probably going to be easier to get that out of the human development part of the Human Cell Atlas.
Well, one thing that we're starting to do quite a lot of is looking at how to combine AI techniques, machine learning techniques, where you're able to extract structure from large tissue samples in automated fashion, but then link that to the physics-based models. So I think, we've seen this enormous surge in interest in machine learning, deep learning, AI algorithms, but we're just beginning now, I think, to explore the relationship between that and the world of predictive computational physics based on the underlying equations governing function. And I think that's a really important step, that we bring those two fields together. Otherwise, AI tends to be just another black box.
Well, I think we probably should try, and we don't have we've got maybe, what, 20 minutes maybe? We should probably try and figure out if we can do anything for this blue box, which they're saying they want some sort of actionable potential working group to carry on or for this. So I think, I don't know, do we even in the green section want to have like an action items or some sort of call it out differently so that we can figure out where to go?
It sounds like one thing that we need is just more data collection in general. So that's certainly one of the action items that we need to work on, I think.
I wonder if in some cases it's data collection or just being aware of different larger data sets. Like we actually weren't aware of the vast clinical and patient image sets that Vanderbilt had until we talked to them earlier in the year, and to realize they have this untapped set of millions of CT scans and MRI scans that just need to be data mined, I wonder if there aren't other collections like that or similar collections. I mean, I also agree that one thing that would be really cool is to use stuff like, I forget what Lightsheet is pretty cool, but I think part of what's hard for that is just the penetration, how deep you can go into the tissue for clearing and things in order to get sort of that 3D data sets, spatially resolved data sets, molecular data sets into that that realm. But sorry, anybody else?
I know we have MRIs for lots of children's data. Vanderbilt probably has that too. But at CHOP, I'm at the Children's Hospital of Philadelphia. We have and I know Penn does too. They have a lot of images. So that would be something I could easily look into for you if I was added to that. And I know as far as things to do, I know that there's active interest, as I mentioned, in the developmental temporal group for doing more work on this common coordinate framework idea for embryos and dynamic growth. So you don't necessarily have to put it in as an action item, but we could bring people in on that action item if they're interested in it, or you could add your own. Or at least talk to the temporal group. Should be some, I mean, there's active computational biologists who, like Dana Pierre, who would like to work on this idea of development. So in dynamic physiology changes.
So just so that I can be capturing sort of like the conclusion to everything in the blue box, is there sort of a problem that you've aligned to at this point to solve? Is it?
I've put some comments into the Google Doc. Can you see those?
Where are you at?
Where did you add them, Peter?
I can't see anything.
Oh, in the blue section?
No.
Oh, no.
Am I putting
Are you
the
in
wrong
document 6B, the role of computation of physiology? Is that the one that people are annotating?
That's the one we should be
okay.
down.
Oh, sorry, I'm putting them into breakout session part 1.
Oh, cool. Well, you're doing my work for me. Thank you.
So one question that comes to me is that it seems like the data's there, and what we need is people power to take advantage of data. So we need tools. There are probably a few 100,000 postdocs here, students who are not quite fiddling their fingers yet, but maybe in a few weeks' time. So one question is, how would we unleash people power to turn what are potentially hundreds of thousands of images, which actually contain useful information, and make it accessible in the context of physiology? Because AI is very useful and we can do lots of things with it, but at the end of the day, I think a lot of this requires a lot of people power.
So I guess in order to turn this recommendation, what we want to do, into a thing, it would be, make the data and the analyses accessible, or make the tools, rather than like, this is what we want, we should word this as make the analysis available. I'll just reword this.
Yeah, please go for it. There's no territory here.
So I guess I view this like the SETI it was a few years ago. People used to want to look for aliens on their computers, scanning the data to a certain extent, but this requires more people input. But if there are, and there'll be tons and tons of scans, the question is, how do you meaningfully turn that into something which is useful and tells us about variability among the human population, etc. And the information is all there, it's just inaccessible and hasn't been analyzed.
I think that, one of the other things that we should do is make sure that we have a comprehensive list of all of the known data sets, so that we know, who has what. Like, just like Ellen was saying that she didn't know that Vanderbilt was doing a certain thing, or now we know, based on what Dee was telling us, that, I think she said Penn has a bunch of data available on kids. That's going to be an important thing for us to collate and have all of that information at our disposal, so that we can then filter through that list and see if there's anything that we can then choose a model organ on, and then develop a scaffold based on that. Does that sound reasonable?
I'm just transferring my comments from the other one across to this one.
Awesome.
It was Penn, right? I'm writing this down.
Yeah, it's the Children's Hospital of Philadelphia. And I'm also at Penn, and they have more adult data. So I'm not sure how open that is, but I know they have a lot of it. So, I mean, I could also check with the Penn people who manage their data, but as far as children's data, we have a lot of MRIs in kids. Now, the thing is about kids, again, they're all sick, as was mentioned earlier, most of them getting MRIs and imaging are sick. The interesting thing is we're going to be getting a lot of MRIs on kids that have unique birth defects as well. So we'll have hearts that have classical birth defects as well as, or other organs that have classical birth defects along with healthy hearts. We should have some healthy heart MRIs and other organs, obviously. I'm just thinking about hearts because that's one of the groups I work with. So that's also an interesting opportunity down the line, is to look at structural differences.
Sure.
We might need again a list of what information needs to be read out from those, images.
Sure. So this would be, institutions which, patient images. Yeah, list of institutions and their associated patient images. That sound okay?
And also like the metadata that goes with that, because you would want to know if there's underlying issues beyond what you're able to see on the image.
Yeah, this should include things like age, sex, probably health status.
Yeah, that can include a lot of stuff, but yeah, that's part of the problem is too, just everybody collects different kinds of metadata, and so that's still an argument as to which things are the most important. But yeah, definitely you need to have some metadata coming, patient metadata or clinical metadata coming with the images for sure.
So Peter, when it comes to things like the mice, how are we doing for amount of information? I mean, in theory, about 20 million mice get used for experiments each year, but it's not clear to me how much of that actually translates back to physiologically relevant information. I mean, surely for a mouse, it should be fairly easy to have a pretty comprehensive set across various types of parameters.
Yeah, I mean, in the Spark project, we are getting on the organs that we're currently studying, which is only 5: heart, lungs, bladder, stomach, colon. But the plan is to go beyond that. We are getting substantial data sets for the mouse in a whole variety of, both vascular, neural, anatomical, cellular level, RNA-seq, etc. data sets. But that'll only be a tiny fraction of what people are generating. But we are at least pulling the data sets we're getting from all these Spark investigators into this common coordinate framework.
If people feel like they're coming to a conclusion on the end of list of that section, I just wanted to mention that there is 2 more sections to go. What at the very end, they're sort of hidden there. What additional expertise do you need, and when the next meeting is? But feel free to continue talking about the recommendations as well, of course.
So, by the way, we've put in a application to the, on behalf of the Spark DRC, we've put in an application to this Common Fund CFDE together with HubMap and Kids First. So one approach, I guess, would be to try and expand that to include more and more of the various projects around the globe that would be able to take advantage of common infrastructure.
I don't know if Peter remembers, but I'm Kids First too. So.
I know that in previous, rather in the other session I was in, we talked about having like cross consortium calls to talk about this. But I know that Katie, she's been Katie Borner, she's been all over the place talking to all these different people about the CCF project, and she's part of HubMap also. I mean, would it make sense to just get another group of us together, like just all of the CCF people that are interested?
I think so. I mean, I think Katie's the right person to be providing the common reference point for this. That she's the one that we've been dealing with HubMap.
Yes. Well, I know basically we've touched with Spark. We've touched we currently have a glue grant with KPMP to kind of figure out more of what they're doing for the CCF. You know, we're with HubMap, of course, and we've been trying to really figure out what components everybody needs in order to really go from tissue to CCF. And I think this has been a much bigger problem that there's lots of details that don't get collected at the beginning. And that's what we've been trying to figure out. And so definitely she's been working with a lot of reaching out to Allen Brain Institute also, just to try and see what is everybody doing for this effort. And so, yeah, definitely, I think she has a very good global view, and trying to bring people together she's very good at. But I think this is a huge thing that has been done not too badly with the brain. That is where a lot of the CCF stuff really started, I think, with the Allen Institute and many other groups. But I think we certainly can learn from them. But there's a lot of new technologies out there that I think can be capitalized on for sure. So
We're also thinking about trying to set up, I'm part of Kids First, like I mentioned, and even though Kids First is more of a genetics project, we have a lot of cancer data that have come from physiological locations. I mean, from anatomical locations that don't have a common coordinate framework or computational aspect to them at all. So that would be, you know, going back, maybe if, maybe one of the action points is, besides me trying to find some pediatric reference data, we can try to come up with at least for the brain, maybe some sort of what was Peter calling them, waypoints? What were you calling them, Peter? It was
Predictable points.
Yeah, the points that we can in the brain, in the pediatric brain, that we could start, that maybe based on the vasculature, that we can start looking at where the brain tumors have been excised from.
Fiducial
First. Yeah, fiducial, thank you, fiducial points.
mhm.
Because that would be something that we could probably do, because we have images on the tumors as well. If we want a project in that direction.
One thing I think it would be nice to capture in the blue box is some relations back to the human cell atlas.
Mhm.
So maybe one of the problems is linking human cell atlas to physiology. I think it's a large problem. It doesn't deal with the details of how we do that, but I think it is a really big super challenge.
How engaged is this group in the biological networks of the HCA? Because I hear these kinds of conversations happening in this context, and you clearly have expertise that you could contribute there.
Yeah, obviously I'm directly involved in the HCA because I have one of the sub-projects. You know, I think, you know, for a whole variety of reasons, physiology just gets missed out because actually the focus has predominantly been, at least in the UK, to the developmental biology. So at least initially was not a disease-focused exercise. So we were, you know, charged with comparing human development with the adult human tissue, which is pretty much an anatomical resolution of the single-cell atlas. But at the moment, yeah, there is just no linkage to physiology. You could argue something with some of the diabetes projects, but yeah. But I think it is something very important because it's a massive community spending vast amounts of money to develop this atlas, rather like they did with the human genome, and that mapping to physiology is, at least from what I can tell, pretty non-existent.
Yeah, and I'll just say I'm not part of the HCA, although I'm part of HubMap and HTAN. I've seen HCA people sometimes at HubMap meetings, but that's the extent of it, really, for me.
Well, so it seems like with HCA, most of the data sets, at least that I've seen, are coming from bulk analysis, as opposed to what HubMap, for instance, is trying to accomplish is spatially resolved data sets, so that where you actually know the location of where the tissue came in a more specific way than just general gross anatomical, and where you would actually have things like spatial transcriptomic data, where you have a slice of tissue from an organ where you knew exactly where that was coming from, and you actually still maintain all of that spatial information because of the methodology that they're doing. Now, there are some bulk RNA-seq type data sets still in HubMap as well, but those data sets are coming actually from more HCA affiliated labs. But I think ultimately, in order to get to where we all kind of want to be, we're going to need to sort of capitalize on combining more specifically where the tissue piece is coming from, and actually utilizing these spatially resolved assays that are being developed. And HCA is, like I said, based on what I've seen so far, not really addressing the spatial aspect as much in terms of maintaining where those cells are located on a slab of tissue. It's been more the bulk data sets. So I don't know. I think they're trying to get to that point, but actually HubMap is sort of, that's their whole goal, is to already start there. So
I think that's changing rapidly. I think a year ago you could do some nice 10x and do some good quality analysis, and you could get an Nature paper. That ain't going to occur anymore. Yeah. And so the questions have to become much cleverer. And some of that may be disease focused, but there's still a lot of chapters of the Human Cell Atlas to emerge, and the spatial aspect will almost certainly be a requirement to get into the journals these PIs aspire to. But yeah, because they're going to need to read physiological function back out, which is difficult in an entirely descriptive 10x type of format.
Yeah.